Hello and welcome. I'm Bernhard Zimmermann from the Business Sector Life Sciences at Zeiss Microscopy, and it's my great pleasure to see you all joining us at 60 nanometers today. I am particularly happy to have with us here today a truly distinguished group of experts in super resolution microscopy. As you will see in a minute, this expert panel will provide you with insights into the recent progress in the field of super resolution microscopy, both from an application and a technology point of view. And based on all of this, you will learn about significant new developments at Zeiss, developments that have once again been geared at pushing the limits of super resolution in order to support the community towards gaining even deeper insights into biological function than ever possible before. So with this, let me welcome the expert panel, Hans Blom, who will guide us through the day today. Hans joins us from Stockholm, from the KTH in Stockholm, Sweden. He's joined by Markus Sauer from the Biotechnology and Biophysics Biocenter at the University of Würzburg in Germany. And the panel is completed by my colleague, a long-term expert and colleague, René Dalrymple, who is calling in from the US today. So before I hand over to Hans, however, I'd like to make you aware that this is not only an expert panel discussion, you will also have the option to ask your questions. And in order to do so, please use the Q&A box that you find at the bottom of your screen in the Zoom window. We will collect those questions and try to answer as many as we can at the end of the session. With this, have fun, and I'm handing over to Hans.
Yes, thank you, Bernhard, for welcoming us all. So yes, very welcome to this event. And as Bernhard said, my name is Hans Blom, and I will be the host of this event. And I'm also very happy to have Markus and René here on the panel. So my background in super resolution microscopy started in 2003, and I have developed STED microscopy and applied that. But since 2011, I am heading a national imaging facility in Stockholm, Sweden, helping users with super resolution microscopy. So also welcome, Markus. Can you tell us about your scientific expertise and background?
Sure. Hi, Hans. Hi, everybody. My name is Markus Sauer. As I've been already introduced, I'm heading the Department of Biotechnology and Biophysics at the Biocenter of the University of Würzburg. And I'm working in the field of single molecule detection and single molecule imaging and the development of super resolution microscopy methods for about 20-25 years.
Yeah, great. Thank you, Markus. So, hi, René. Most welcome. Tell us about your background and expertise.
Hi, Hans, and hi, everyone. I'm really excited to be here with you today. So as Bernhard mentioned, I am currently a product marketing manager with Zeiss in North America. But prior to that, I spent many years as a super resolution specialist and actually got my start with microscopy working with home built systems for doing single molecule localization microscopy.
Yeah, perfect. So maybe we are a good panel knowing about super resolution microscopy then. So let me then start to share my view about what users in my facility ask for. And the first obvious thing is, of course, they want to come and get more resolution. And here, this is exemplified by looking at fixed neurons. So by resolving these with SIM, STED, or Palm, we can then, of course, see things better. And the second thing users ask for is they want to see things in multicolor, label many different structures. And this is exemplified here with multicolor of synaptonemal complexes to the left, then resolved with SIM. And to the right, you see an example of focal adhesion. And there it's also resolved then with dual color STED or dual color D-Storm. The third thing users ask for is, of course, they also want to improve 3D imaging. They want to have better resolution in the axial direction because biology is really 3D. And we can provide that, as you can see here to the left, there's 3D SIM imaging of bacteria. In the middle, there is 3D STED imaging of mitochondrial protein. And to the left, there is 3D palm imaging of spine structures in neurons. And then the fourth thing users ask for is, of course, they want to see more. They want to see a lot. And here it's D-storm imaging on our system where we see about 20 microns. If we then switch to SIM, we can see 50 microns and still resolve more. So they are always asking for seeing more and bigger field of view. And the fifth thing that users ask for is live imaging. Can we do live super resolution imaging? And this is exemplified here with steady imaging of actin rings. And then they also want to go into thick tissue, exemplified here with the cochlea, the inner ear. And if that is decalcified and sliced, we can then, for example, image connexine sheets with SIM microscopy. And then the last thing I want to share with you is speed, of course. They want to measure very fast, live 3D. And in our facility, we have had an in-house collaboration with Sara Abramson to merge multifocus microscopy with SIM, so we can get an instant 3D stack of cell structure with high resolution. But we would like to have even more speed or even more resolution, or actually a turnkey system that we can apply in the facility. So that will be our dream. So Marcus, what about your insight regarding this? What's your need as a scientific leader?
Thanks, Hans. I'm also heading an imaging center here, the Bioimaging Center of Würzburg, and I can only confirm your assessment of the current needs and wishes of imaging users and questions they have. So today we have different super resolution microscopy methods at hand, including D-storm, palm, STAT, image rescanning or array scanning microscopy, structured illumination, and different correlative super resolution microscopy approaches. But all methods have limitations, even though they allow us to bridge the resolution gap between the atomic resolution here provided by cryo-EM on the left-hand side and whole cell imaging by positron emission tomography. They still have limitations. Either they are limited to fixed cells and complex instrumentation, requiring expert knowledge and are restricted to a few dyes that work perfectly only, or they do not achieve sub-100 nanometer resolution. For example, on the one hand side, we have single molecular localization microscopy techniques like D-storm and palm microscopy that enable imaging with virtually molecular resolution by temporally separating the fluorescence emission of individual fluorophores by either photoactivating, for example, fluorescent proteins, or photo switching of organic dyes by adding a switching buffer, so-called photo switching buffer, and which is finally followed by image reconstruction. Here, the precision, localization precision we can achieve is mainly determined by the number of photons detected. And this means that we can today achieve localization precisions of only a few nanometers, let's say of 5 nanometers, as shown here for the actin mesh work, using our favorite dye, Alexa 647, labeled to phalloidine, which binds to actin filaments and allows us to visualize the details of the cytoskeleton here. And the other example shown on the upper left is the nuclear pore complex, which we introduced 2012 and is used still today in most labs as a reference structure for to show the quality of different new super resolution microscopy methods. Yeah, but the spatial resolution since then has not been substantially improved. Yeah. The next example I want to show here is that you can also use this single molecular localization microscopy techniques like D-storm advantageously to investigate bacterial infections, like shown here, bacterial infections of endothelial cells with Neisseria meningitis invasion sites. So here we labeled a ganglioside, a lipid in the plasma membrane of these cells, the ganglioside GM1 with choleratoxin B, which binds to GM1, labeled with Alexa 647. And in uninfected cells, so this ganglioside is homogeneously distributed in the cell membrane. However, upon addition of these bacteria, which express GFP in this case, we see that these gangliosides accumulate around this bacterial sites. So just in this way, facilitating bacterial entry into the cells. Well, However, such high spatial resolutions are so far only obtained when imaging fixed cells, and they work usually only with a few dyes, as for example, D-Storm with our favorite dyes, Alexa 647 or Sci5. And the experiments require expertise, and quantitative experiments are restricted to a single color usually. Furthermore, they require high irradiation intensities of several kilowatts per square centimeter, which damage cells. Therefore, we also installed a few years ago a Lattice light sheet microscope, which has been recently introduced by Eric Betzig at Genelia Farm and his colleagues. And this allows us then to visualize in live cells dynamics of ER and tubulin, as shown in these videos here, and taken on our Lattice light sheet microscope. And we could visualize then all the dynamics which have been published already, for example, in the seminal papers of Jennifer Lippincott-Schwartz and her co-workers in the Cell and Science paper. However, the resolution in these experiments, even though temporally very high, is spatially restricted still to a few hundred nanometers. Of course, you can further increase resolution by combining it then, Lattice light sheet microscopy with D-Storm. But then you have to fix cells again and visualize, for example, the distribution of plasma membrane proteins like here, the receptor CD45 in the plasma membrane on the apical and basal side, but also at the interfaces between two cells. This is, of course, very interesting, but this is, again, restricted then to fixed cells if we want to go for a higher resolution. So to conclude, under practical usage, thus, a gap exists between the 10 and 50 nanometer resolution provided by D-Storm and STAT microscopy, for example, and these methods are mainly restricted to a few dyes and the addition of special buffers, and structured illumination microscopy on the other hand side, which achieves the resolution of only about 100 nanometers. So a method that enables 3D multicolor super resolution imaging in this 50 to 100 nanometer spatial resolution gap, yeah, with standard fluorophores and fluorescent proteins ideally in fixed and living samples, and with a high axial section probability for tissue imaging combined with a high temporal resolution, yeah, would close this gap and find widespread applications in biomedical imaging. But since we don't have these techniques at hand in the moment, we thought 2 years ago to combine now super resolution microscopy, at least for fixed cells, with expansion microscopy. Expansion microscopy has been introduced by Ed Boyden's group 2015 in this Science paper. So basically you link your protein of interest into a charged hydrogel, you digest the proteins or you denature so that the intermolecular protein interactions are destroyed, and you finally expand, and by leaving the sample overnight in water, and this results in swelling of the sample 4 to 20-fold, typically depending on the protocol. And this way now we can label 3 proteins in this synaptonemal complex. Maybe you remember the helical images Hans showed in his introduction. We can now do 3 color experiments. We use 3 to 4-fold expanded synaptonemal complexes, label them after expansion, and then do structured illumination microscopy. And then we end up with a resolution of about 30 nanometers with 3 colors. And we can really resolve details of the molecular organization of this multi-protein complex, which have previously not been possible. An example is shown here, for example, a striking morphological feature of the lateral elements of this synaptonemal complex here, the protein Zeiss CP3, which could not have been resolved before by light microscopy, is the occasional splitting into homosub lateral filaments, as shown in panel G here. For example, here, if you see my cursor, and on the right-hand side, for example, in electron microscope images, and above the fraying at the ends of these lateral filaments. We could resolve thanks to the spatial resolution of about 30 nanometers we achieve in these experiments. Yeah. However, expansion microscopy works again only with fixed samples, and the preparation steps are time-consuming, and control experiments have to be performed that demonstrate isotropic expansion of the sample, and these are really annoying, these experiments, and sometimes really challenging, I can tell you. Hence, it would still be desirable to have a method at hand that enables imaging in the 50 to 100 nanometer range, even in living cells.
Okay, great, Marcus. Thanks for your broad scientific view on what more is needed. So, Renee, what is Zeiss' view on this customer need regarding super resolution microscopy?
Sure. So thanks, Hans. It's really interesting to hear from both you and Marcus about what you really need to complete your research. And that is exactly what we try to do here at Zeiss as a microscope development company, is really stay in touch with the research community to understand where the needs are, where the gaps are, like you were discussing, Marcus, so that we can do our best to fill them with a turnkey commercial solution. So to do that, in addition to speaking with people like yourselves, we put out broad-reaching surveys to understand what people's interests are. You can see an example of that here on the left, where we saw that there definitely is a very large demand for live cell imaging. And also, if we look into the publications in the research community, we can see that the combination of live cell imaging with super resolution microscopy is a very growing field, and it just continues to grow over the years, as you see in the graph on the right. So certainly this is an area where there's a lot of need, and we can learn a lot more about our biology by going into it. So to that end, we're always trying to think about what the best technology is to really develop and commercialize. So in order to do that, we thought about structured illumination microscopy, of course, because it really is one of the most live cell compatible super resolution techniques. At this point, it's also a very, very well established technique. You can see in the graph on the right how much it's grown over the years and how many times it's been cited in all of the publications. And really, I think it's widely accepted in the community that structured illumination microscopy is extremely well suited for live cell imaging as far as in comparison to other super resolution technologies that are out there today. And you can see that here in the quote from Eric Betzik and his collaborators, where they identify that SIM stands out because of its ability to image with high speed and at low illumination intensities. With structured illumination microscopy, we're able to achieve currently a twofold improvement in resolution in all three dimensions. So that really allows a lot of access into biological structures that weren't previously able to be seen with live imaging. It's easy to approach because it has standard sample preparation requirements, which means that you don't have to use any special fluorophores, and multicolor imaging is really easy. Additionally, because it's built on a wide field platform, we can achieve really nice large fields of view very quickly in order to get as many statistics and as high throughput as possible. So of course, I took a look at this and decided that structural illumination microscopy was the way to go and was definitely going to grow in the future. So let me take you just a moment to back up and explain a little bit about what structured illumination microscopy is and how it works. So with structured illumination microscopy, we used patterned light or structured light, which we place on top of the sample, and we're able to use it to extract high frequency information from the sample, meaning the high resolution information, the fine details that you want to see. So for example, on the right here, you see a wide field image of actin. And if we image this same cell with super resolution SIM, we can see the much finer details in the sample that we achieve with that twofold improvement in resolution. But how is that done? So let's take a look at this cartoon and pretend for a moment that these parallel lines here are, let's say, microtubules. Of course, they don't have to be parallel, this is just a cartoon. But what's important is that they are spaced so close together that they actually cannot be resolved with the typical microscope because they're at a high frequency. Now with structured illumination microscopy, what we do is we take a patterned light and we shift that across the sample. You can see when we do that, that there's the formation of these dark squiggly lines. Those lines are called moire fringes. And the important thing about moire fringes is that they're actually spaced far enough apart that we can resolve them with the microscope. So if we can resolve the spacing of the moire fringes, and we know the spacing of the patterned illumination that we put onto the sample, it's a known frequency, we can essentially use that information to solve a very simple linear algebra problem to extract the high frequency or high resolution information from the sample and see how far apart in this example those microtubules were. So in a nutshell, that is how classical implementations of structural illumination microscopy work. And in those classical implementations, we use a stripe pattern like you see here. Now, a couple of years ago, Zeiss really wanted to push structural illumination microscopy even further. And in order to do that, we developed different optics which allowed us to change the pattern of the structured light to utilize a three-dimensional lattice pattern rather than a striped pattern. And this is a little bit about what that looks like here in 2 dimensions. But you can see that as it shifts across the sample, it works in the same way that we still get the formation of these moire fringes that we can resolve, and we're still able to calculate the super resolution or high frequency information in the sample. Now, shifting to a lattice pattern instead of a stripe pattern comes with a number of benefits. When you have the classical implementations with a stripe pattern with SIM, in order to get super resolution in all the different directions to have a more isotropic image, you actually need to shift the pattern across the sample, and then you have to rotate it, shift it again, rotate it, and shift it again, at least 3 times, if not more. Now, what this means is that we end up exposing the same area of the sample to light multiple times, which of course leads to excess illumination, which means we have more phototoxicity and bleaching. And of course, we always want to minimize this. It also takes some time to stop and rotate the pattern. With Lattice SIM, the dimensionality is already encoded in the pattern, so there's no more need to rotate it, thus allowing us to be more gentle but less light into the sample and not take the time to rotate the pattern. Additionally, the pattern affords us better signal to noise in our image and better what we call modulation contrast. You can see that in the graph on the right. If we were to draw a line profile through the stripe pattern and the lattice pattern, plot the intensity over the line, that's what you see on the right, you can see how much more clear the pattern is with Lattice SIM because of that better modulation contrast. What this really means for you and for your imaging is that you're able to get higher quality images with the same laser power dosage. So for line imaging, you can turn the laser down and image more gently. Or if you happen to be imaging into a thicker, more scattering sample that conventionally was challenging for structural illumination microscopy, you're able to penetrate significantly deeper into it and still have that 2-fold resolution improvement. So if we take a look at some application examples for what Lattice SIM enabled, we see that it also allowed us to push the speed of acquisition, which of course is so important for live cell imaging, all the way up, in fact, to 255 frames per second. So we can capture things like this dual color image of endosome zooming around in our sample. We can do so gently because of that modulation increase, where we're able to turn the laser power down and image longer. We can push deeper into scattering challenging samples, such as this uncleared brain tissue section that you see at the bottom left. And of course, we're able to always get better image quality because of that modulation contrast improvement as well, while still maintaining the benefits of classical SIM with easy multicolor imaging and a large field of view.
Yeah, looks great.
Thanks, Hans. Well, I know that you recognized with your SIM instrument that you wanted to push it even further, and of course, so do we, as well as do a number of members of the community. So there have been some innovations in structural illumination microscopy over the years. You can see a sample of them here on the slide. And a lot of these techniques, they try to push SIM in regard to resolution, speed, gentleness. The challenge with a lot of these implementations, of course, is that, such as the multi-focus SIM that you worked on, which is a fantastic innovation. However, you had to build it yourself in your core facility, right? And what we want to do is really provide access to these types of technologies to the widespread community so that they're easy to use to push our research further. We also want to, of course, try to improve upon all 3 of these categories at the same time, rather than having to sort of give one up for the other.
Okay, great overview of Lattice SIM then. But today we are, of course, all here to hear about something new, that you have new possibilities with Lattice SIM. So Renee, please reveal to all of us your new innovation. I am very curious.
I'd be happy to, Hans. So first of all, I want to really thank all of you for joining us today, both Hans and Marcus, and everyone else who's called in with us. So you may have seen this graphic here on social media or in your invitations, and I'm sure you're all curious on how exactly we're going to be joining all together here at 60 nanometers live. But so I'm very happy to announce today that in order to help us all reveal the vibrant suborganelle network of life, we are introducing today a new technique called SIM². And if we take a look at what SIM² can achieve, we have here a simultaneous dual color image of the ER and microtubules. And we can see in this nice large field of view on the left, if we zoom into a tiny region there and take a look, we can really see how these structures are interacting with one another. And if we pause the movie for just a moment and take a look at the resolution that's achieved in this live image, we can see here that we're easily able to achieve resolution beyond 100 nanometers in a live sample, even all the way down to 60 nanometers, which we can see here in these line profiles on the left. So this should really open up new opportunities in gentle live cell imaging at the resolutions beyond 100 nanometers, which of course, Marcus, is the gap that you were mentioning would be extremely helpful to fill. Additionally, because we're looking at multicolor imaging, because there's no special sample preparation required, we can also, of course, try to understand how these different structures interact with one another and how they're related spatially. So here you see an example where we can measure the distance between the two structures quite precisely. In fact, even beyond the 60 nanometer level when we're talking about multiple channels. So let's take a little bit of a closer look at the improvement of SIM² versus conventional SIM implementations. Because of course, we all know that there's more to see beyond 120 nanometers. So if we take a look at some microtubules here shown in wide field, of course, with wide field microscopy, this is sort of what the raw data looks like for SIM. We see a lot of out of focus light that are sort of hiding our structure that we really want to resolve. With structural illumination microscopy, we get a twofold improvement in all three dimensions already, and you can see a drastic improvement here. However, there's more to be seen. So with SIM² now, we're able to double that resolution again, get down to 60 nanometers. And here we can quite clearly see the microtubules actually twisting around one another and measure the gap in between them. So I want to point out that, this technique truly is revealing what is really there with our super resolution capabilities, and is not just, an algorithm that is artificially narrowing our structures, in which case we wouldn't be able to see the gap there in those twisting microtubules.
Yeah, thank you, Renee. That's quite impressive. Nice images.
Thank you. Yeah, we're really excited about it.
So I know, Marcus, that you have tested the system. What can you say?
Well, yes, we had the chance to test the system, but the first image I saw, it was a SIM² image of an actin skeleton I saw a few months ago. And to be honest, I couldn't believe that this is a SIM image. It looked so close to a D-Storm quality image, is the resolution, that we couldn't believe it. And this at a speed with 3-4 color options. So we were really, we were impressed really about these images, I have to say.
Okay, yeah, very nice to hear then. But I have not tested this system, so I don't really know what goes on. So what's the magic behind this?
Well, there's no magic, Hans, I promise. So it's all soundly based in math. Do you have any guesses? I know that, I saw a lot of buzz on social media where people are trying to hypothesize what we might be doing to get to 60 nanometers. What do you think?
Well, I can have a guess. Maybe you have some machine learning algorithm that process the images to enhance the resolution. I don't know.
Well, I'm going to tell you here in just a moment. But no, we really wanted to focus on extracting as much super resolution information as we could from the images we collect and utilizing information to the fullest. So meaning we wanted a technique that was going to be widely applicable to all different kinds of structures, even those that haven't been discovered yet. So there's no need for a training data set or a priori knowledge about the sample structure. We're really extracting information from the images we collect. So let me tell you a little bit more about how it works and sort of ease everyone's curiosity, because it's extremely important that you all understand exactly where your data is coming from and how the images are coming to be. So what do we need to do to optimize a SIM image? Well, there's sort of two parts of this. Of course, the first part is the acquisition. And really with the introduction and development of Lattice SIM, we really focused on optimizing the optics and the pattern that's utilized in order to collect as much information as possible. The second part of this now is taking everything we learned from Lattice SIM and all the information that it provides and utilizing it to the fullest through optimizing the Sim Reconstruction process through the introduction of SIM². So if we think about how a conventional Sim Reconstruction works, of course, we're going to take an image of our sample with our patterned light. We're going to shift the pattern and all of that. And then what we're going to do is see the information from those moire fringes that we talked about earlier and extract the high frequency information and shift it back to where it belongs in the image. Now, in order to do that with classical SIM implementations, this is all done in one step where we utilize a point spread function from our microscope that was collected, and we combine that as well with a deconvolution step. Okay, so this all happens at the same time. Now, with the introduction of SIM², what we're doing is really teasing apart the different steps in the Sim Reconstruction process and optimizing all of them so we can utilize the information to the fullest. So we're still going to take our image in the same way, and we're going to go through our Sim Reconstruction process. And now in parallel, what we're going to do is also optimize the point spread function that we're using into something that we call the Sim PSF. The Sim PSF goes through all the same steps that our image information does in our Sim Reconstruction algorithm. This information is then fed into an iterative deconvolution step in a second step rather than combining it all in one, so that we have the opportunity to really optimize everything. So let's talk about deconvolution for a moment. Deconvolution has been widely used in the literature for many years now. And in fact, there are many publications out there discussing the fact that it allows frequency restorations beyond the frequency limit of the raw data that's collected. Now, how well you can do that depends on a couple of factors. It depends on how well sectioned your image is, how well you know the point spread function of your microscope, and of course, what the signal to noise ratio is in your data. So if we think about applying deconvolution to different types of imaging, we could start off thinking about wide field microscopy, where deconvolution is used quite often. You can see here that we do, of course, get a resolution improvement. And that's because we're able to measure the point spread function of our microscope fairly easily. And you have pretty nice signal to noise ratio, because in wide field, you're collecting all of that light. Even the out of focus light is utilized and reassigned back to where it originated. When we apply deconvolution to a confocal image, we also see an improvement. And that's because we have a nicely sectioned image, since you're using your confocal pinhole for optical sectioning. You can also, of course, measure your point spread function. However, we don't always have the same signal to noise ratio that we do with other techniques, because we're actually rejecting some of the light from the sample by using that confocal pinhole. This gets even worse if you start trying to close down your pinhole smaller and smaller to improve resolution. Now, what about structured illumination microscopy? So a conventional SIM, as I mentioned earlier, the deconvolution step is part of the Sim Reconstruction already. So if we combine all that together, we get a twofold improvement in resolution. So that's our 100 to 120 nanometer limit we're all familiar with. And this is because we do have sectioning, we can measure our PSF, and SIM provides pretty good signal to noise. It's based on a wide field technique. But what if we could push these three parameters even further? And that's exactly what's done with the new SIM². And this allows us to achieve an additional twofold resolution improvement over conventional SIM so that we can reach that 60 nanometer level. So what really enables this? Well, it's two things. It's the optical improvements that were made with Lattice SIM illumination, because again, that really allowed us to achieve better sectioning and higher signal to noise ratio because of that increased modulation contrast and all the information that is encoded when we collect our Lattice SIM image with that pattern. The second part of it is really to optimize the processing step, as I mentioned with SIM², where we're really looking at the point spread function and making it as optimal as possible with our SIM PSF. So if we take all of this together, what are the benefits of doing it? Well, I've already talked a lot about the resolution improvement down to 60 nanometers laterally, but we also see a really nice improvement axially. So you can achieve sectioning to under 200 nanometers, which is just incredible. Additionally, we're able to increase the speed of acquisition by really fully utilizing all the information we collect with Lattice SIM and the SIM² algorithm. And we're able to make it more gentle by reducing the phototoxicity. So I'll talk a little bit more about how that works in a bit. Another thing I want to mention is that our SIM² algorithm is not only applicable to the full Lattice SIM implementation, but also for the SIM² epitome mode. Now, if you're familiar with our Elyra 7 platform, when we launched that Lattice SIM, we also launched something called the SIM epitome mode. And what this does is also uses a structural illumination pattern, but it uses lower magnification objective lenses. So anything from a 10x to a 40x. So you can get increased resolution and really fantastic sectioning with much larger fields of view. So SIM² now is going to take that mode to the next level as well.
Okay, thank you, Renee, for telling us about the details. So very nice development. So this was developed by Zeiss, and now you allowed Marcus to play around with this system and test it. So can you share?
We always want to see how it works in real labs, right? With your samples. That's the whole goal, is to advance your research as far as possible.
Great. So Marcus, share your views, experience.
Yeah, we had the chance to test the new Lattice SIM² in my lab, and we were, as I just mentioned, fascinated by the possibilities the method provides. So it's easy to use. Standard sample preparation methods, even fluorescent proteins, can be visualized in living cells at super resolution, and it's super easy to handle and really, really fast if you are used to the speed of the older structured illumination microscopy versions. So you prepare your sample as you are used to prepare your sample for standard confocal or standard SIM measurements. There is no buffer required or no special additives have to be added. And you start your multicolor imaging experiments and fix the living cells with high frame rate, and finally let the SIM² algorithm do the job. However, we were still skeptically a bit. I mean, therefore we thought about, let's test the resolution by again imaging the synaptonemal complex, as I just showed you before, where we did this 3- to 4-fold expansion and then put the sample on a structured illumination microscope. So we did these experiments together, again, together with Ricardo Benavente from the Biocenter here at the University Würzburg. And to cut a long story short, I mean, what you see here are 3 color images, and they exhibit the same quality as the SIM images after 3- to 4-fold expansion. Yeah. So multicolor imaging of the synaptonemal complex by Lattice SIM² allowed us to achieve a resolution of about really 60 to 70 nanometer in lateral direction. And this allowed us to resolve fine structural details of the molecular organization of this important multi-protein complex. So it allowed us, for example, to resolve the distance between the C and the N terminus of this transversal element protein shown on here on the left-hand side, Zeiss CP1, shown in the panels here in red and green. So we used antibodies directed against the C and another antibody directed against the N terminal end of the same protein, and we could really visualize the overlap of the C terminus with the central element and of the N terminus with the lateral element. So this we could never achieve before or show before. And this on an unexpanded sample. And here, just for comparison, we could also resolve these hidden details like the fraying at the ends. We're using SIM² of unexpanded samples, or the splitting of these into lateral elements as shown here and also seen in electron micrographs or in standard SIM images after 3- to 4-fold expansion. Yeah. Of course, if we talk about resolution, another sample we usually take is then we use clefering-coated pits, which have a known diameter of about 80 to 120 nanometers. And if you can really visualize these CCPs as hollow bodies, as you can do it on a SIM², on a Lattice SIM² microscope, you can be sure that you really achieve dramatically sub-100 nanometer resolution as we really can reveal these hollow structures with membrane to membrane diameters of about 80 to 100 nanometers. But we also tested the sample on a highly topical sample. This is a SARS-CoV-2 infected cell. And we visualized, for example, as shown here in 3D, the amplification of the viral RNA inside of the infected cells by RNA fish and using 3D Lattice SIM². But how is it with live cell imaging? Of course, this is the final goal of all these technologies. So to test live cell, we transfected cells with calreticulin TD tomato and imaged then, because this is a marker of the ER, we could image and visualize the dynamics of the ER with a frame rate of 255 Hertz. And for comparison, I show here just snapshots here taken by standard SIM and in the middle SIM², and on the right-hand side, some details view, and you clearly see the higher resolution. How is it about two-color imaging? So we co-transfected the cells then with EMGB, GFP expressing an microtubule binding protein, and we could then visualize the dynamics of the ER and the tubulin together at a frame rate of 81.6 Hertz. And on the last slide I can show here, I show you some example videos where we see on the left videos on the left-hand side splitting and growing of microtubules, and on the right-hand side in this video we see ER hitchhiking events on single microtubules with really high spatial resolution.
Okay, thank you, Marcus. It looks great. Nice images. So, René, you wanted to explain a bit more about the improvements, what you can do on the new Lattice SIM² platform.
Yes, well, first of all, Marcus, let me just say that is so exciting to see what you've been able to do with this and how it's able to really, hopefully make your life easier by not having to expand your samples to see what you need to see. So thank you so much for sharing.
You're welcome.
So yeah, I mean, we've talked a lot about the lateral resolution improvement down to 60 nanometers, but I wanted to sort of take a moment to also show you the sectioning capability as well as the improvements in gentleness and speed. So what I have here is a side-by-side Z-stack of conventional SIM versus SIM². And what we can see here is how well SIM² is able to deal with suppressing out of focus light to get the thinnest sections possible. So if we play this movie, what we see as we move through the Z-stack is that the out of focus light that's still present in the SIM image, because you have a little thicker section, is nicely split out in the SIM² image. Now, on first glance, maybe you'll look at that and say, well, René, it looks like there's some information missing. But no, in fact, it's actually just sectioned much more thinly. And we can see that if we take a look at the maximum intensity projections side by side, that all the information is, of course, there as it should be. And we can also see here the lateral resolution improvement as well. So I mentioned that the SIM² algorithm also works for our SIM epitome mode, so that you can achieve these kinds of results with a lower magnification objective lenses as well. So if we compare here the field of view that you get with the SIM² epitome mode on the left using a 40x objective versus the Lattice SIM that we had previously on the right with the 63x objective, you can see the much larger field of view. And what's really, I'm so excited about is if you zoom in here, you see that the image quality between the two are nearly identical. So you're now able to reach nearly to the resolutions that you could with conventional SIM implementations with the SIM² apotome mode, while also collecting information on a much larger scale. So more statistics, higher throughput. I know, Hans, you mentioned this is something that your users are always asking for. They want a larger field of view. So what do you think? Is this going to help them in their work?
Yes, it looks quite impressive, actually. They always want to see more.
Yeah, we all want to get more statistics, right? So we can spend less time at the microscope. So additionally, in addition to increasing both the resolution and the field of view with the SIM apotome mode, we are also able to image faster now and more gently because of all the information that we're utilizing from our Lattice SIM acquisitions. So what I mean by that is, how we said it's required to take multiple exposures in order to collect a single super resolution image, because we just shift the pattern over the sample. But what we've done is reduced the number of exposures that are required in order to get to that final image. So rather than collecting 13 images for a full-fledged Lattice SIM², now we can do it with 9, while still maintaining our super resolution capabilities. Additionally, if you're familiar with the Elyra 7 with Lattice SIM, we also have a modality called the leap mode. The leap mode allows us, when we're collecting 3D data, taking Z-stacks, to actually only acquire every third plane and reconstruct all the intermediate planes. So we can get much faster volume imaging while exposing our samples to less light. If we combine that leap mode with this reduction in necessary phases or exposures, we're able to get a 3-to-1 ratio between exposures of our sample and final super resolution images for the full-fledged Lattice SIM² mode. We're seeing an example of that here on the left, where we're looking at the ER. This is a 3D image where we're looking at a maximum intensity projection. So this is going to allow us to image even faster and more gently. So this works not only for Lattice SIM, but also for the SIM² apotome mode that I spoke of. Now, this is even more exciting to me, I think. So the SIM apotome mode already requires fewer camera exposures in order to collect the full image. So instead of collecting 5 images as we did previously, now we can get that down to 3. What this means is if we combine this with the leap mode, we now have a 1-to-1 ratio between exposures of our sample and final super resolution images when we're imaging in 3D. So that means you have completely lossless imaging, and you're essentially able to capture at the same rates and light dosages as wide field microscopy, while achieving resolutions that are nearly comparable to what you could previously do with conventional SIM. So I'm really excited to see what this does for live cell imaging over these really nice large fields of view as well.
Yes, nice development.
Thanks, Hans. I hope that helps your users in your facility. So I want to also mention, I've shown you a lot of cell culture examples. And of course, we want this to be a much more flexible tool than just looking at cell culture. So here we have an example of a large brain tissue section. This is an uncleared section. And with the versatility of being able to utilize different magnification objective lenses from a 10x to a 63, we can take a nice large overview image, find the area of interest. Here we've moved to the 25x lens, where we can already resolve neuronal spines. Then we take it up to the next level with the 40x lens, and then all the way to the 63, so that we can get down to that sub 100 nanometer level, even deep into a scattering tissue sample. So we can really resolve the fine details in the neuronal spines and measure things like spine necks for quantitative imaging into a scattering sample. To show you a few more examples, we worked with a number of different beta testers in addition to yourself, Marcus, and they really put the system through its paces with all these different samples. So here we have examples of C. elegans on the upper left, multicolor imaging. On the top right, we have a zebrafish embryo where we were able to collect a 100 micron thick Z stack. So that's what you see in the upper right-hand corner is a color-coded projection. The bottom left, we have Drosophila embryos where they wanted to do an overview with low magnification, find the area of interest, and zoom in with higher resolution. And then on the bottom right, we have an example of a very challenging sample traditionally for SIM, plant samples, where the researcher was really excited because they were able to penetrate 3 cell layers into the sample without having the challenges that you normally would with scattering and chlorophyll and autofluorescence and everything you run into with plants. So hopefully this gives you an idea of sort of the breadth and versatility of the system and how it can be applied, not only to looking at the nitty-gritty details down to 60 nanometers in cells, but also these much larger model organisms as well.
Yes, very impressive images and a very versatile platforms, it looks like. So how can I now get hold of this Elyra SIM² platform?
So the new SIM², Hans, is available on our Elyra 7 platform, which is our dedicated super resolution platform, which of course is capable of so much more than that now. So with the addition of SIM², we're able to achieve that 60 nanometer lateral resolution, sectioning down below 200 nanometers axially. We're able to push the speed and the gentleness significantly again, even further than we did with Lattice SIM a couple of years ago. And of course, this also provides robustness, so imaging into those thick scattering challenging samples. So you can see even more examples at the bottom of the slide here that I didn't have time to get to today, but it ranges all the way from looking at bacteria, doing multicolor imaging so you can tease apart whether a protein is on the inner or outer membrane, looking at trafficking of fast-moving organelles in the sample and seeing how they interact, photosensitive samples like yeast, which are conventionally very challenging, and all the way up to those larger samples such as plants and zebrafish, et cetera, that I mentioned. So with that, the Elyra 7 platform truly becomes a very versatile tool to hopefully help everyone push the boundaries of their research into areas that, previously we had that gap that really needed to be filled. So it allows us to maintain all of the benefits of the classical SIM implementations with a nice large field of view, pushing it even further with the utilization of low mag objectives with the SIM² epitome mode. And of course, the ease of use that you expect of not having to utilize any special sample preparation, standard fluorophores, and really being able to approach samples on all of these different scales.
Yes, looks very nice.
Well, we're really excited to see what it does for your core facility, Hans.
Yes. So thank you, Renee, and thank you, Marcus, for sharing all your knowledge about the SIM² platform. And I'm sure that there will be several questions here from the audience that they would like to ask you. So maybe we should start the Q&A session here. So there is a question here for Marcus. How easy it is to use this system? How long do you have to train me in order to use it?
If you're familiar with the structured illumination microscope and with the Zeiss software doing SIM data analysis, it's straightforward. There is no training required. Yeah, basically.
Okay, so it goes quite fast. I just use my old sample and then the iterative deconvolution is in the Zen software.
Yeah, you can play around with it until you're satisfied with your results. That's basically identical to the standard SIM procedure algorithm and data analysis.
Okay, yeah, perfect.
Yeah. Hans, we know that iterative deconvolution can be a little bit fiddly sometimes, but we really worked hard to set some really nice default parameters, based on whether you're working with live samples or fixed samples in the signal to noise ratio, and try to make it as automated as possible. And then, of course, for those users who really want to get in there and play with the nitty-gritty of the parameters, that's all accessible as well if desired.
Okay, sounds perfect. So there's a second question here then for Renee. It's how live cell compatible is the method? Have you measured for very long times on these systems?
Yeah, so I mean, that's what it's all about, right? Is trying to fill that gap into live cell super resolution at a whole nother level. And so, of course, we've done some tests. We showed some of them here today. But to give you some examples, I know we also worked with looking at actin ruffling, which is sort of a nice benchmark for cell health to make sure that the cell is behaving as it should. And we imaged continuously with a Z-stack, collected over 15,000 images without seeing any appreciable photobleaching or phototoxicity, which means we could have imaged even further. So we're really quite excited when in the realm of super resolution, right, of really being able to push this even farther than we could go before. Now, that being said, if you're trying to push to imaging for days at a time, right, collecting 1 million images, that's where something like the Lattice light sheet technology comes in as truly the most gentle. However, it really comes down to how much resolution you need out of your sample, sort of which is going to be the best choice for your application.
Okay, yeah, sounds very good then. Very live cell compatible and super res. So yeah, there is this, of course, question. You started a bit talking about it, that what artifacts can there be then in this iterative deconvolution process?
Yeah, so as I mentioned, optimizing the SIM² algorithm, one of the really nice benefits actually is that we're able to pretty much eliminate the conventional artifacts that you see in classical SIM implementations, especially when your sample starts getting more challenging. So those are pretty much gone. And of course, what's left is what you would typically see with an iterative deconvolution approach if you were to sort of push it too far or your parameters aren't quite right. But to that end, as I mentioned, we really work to optimize the default settings. And if necessary, you can go in and tweak them further if you'd like.
Okay, sounds like a robust system then. So regarding imaging and collecting imaging and reconstructing the images, how fast is this process?
Marcus, do you want to comment?
As fast as with the standard Sim Reconstruction algorithm. We didn't see a big difference there.
Okay, so it's done in just like 30 seconds or a minute or?
A minute or so.
So of course, it depends how large your data set is, but it's GPU enabled to really push the speed.
Yes, there is of course Elyra users out there in the community, and they of course now ask, can you add on this SIM² modality on your old system?
Yeah, so for existing Elyra 7 users with Lattice SIM, it's a very easy upgrade to add this capability to your system.
Okay, good. And there's a question here, of course, how thick can you image, or how thick do you dare to image with this platform?
Sure, so I showed a few examples into thick scattering samples. We certainly already know we can image 100 microns into uncleared brain tissue with excellent resolution of neuronal spines. And we're pretty confident that with the improvements in the SIM² algorithm, we could potentially push that even further depending on the sample, especially in like a cleared sample or something, for example.
Okay, very impressive.
Yeah.
So regarding this modality, Lattice SIM and Lattice Apotome, what's the maximum speed you can achieve on the system?
So Marcus gave us a nice example of some live imaging at 255 frames per second. So essentially you're just camera limited at that point with the speeds that you can achieve. And you can even do that with 2 colors simultaneously with the dual camera.
Okay, very nice. Yes, so final question here regarding maybe dim sample that has been a problem maybe with classical SIM. Do you have any input here?
So what I would say essentially is whatever image quality you were able to achieve previously with classic Sim should be improved with the introduction of the SIM² algorithm.
Yeah, this I can confirm. If the quality or the brightness of the sample was good enough for the standard Sim, it also works for the Lattice SIM².
Yeah, it might even be improved, and you can even push to dimmer sample. So yeah, very nice. So thank you, the panel, for sharing all your knowledge, and the audience for listening in. So questions that were left there will be answered by Zeiss on their homepage. And now I would like to hand over to Bernard to end this session.
Yeah, thanks a lot, Hans. This indeed brings us close to the end of this session. I don't want to let you go, however, without thanking you, especially Hans and Marcus, for engaging in this event with us today. So big thanks for that. Marcus, an additional thanks to you for having us at the beta test. I'm pretty sure that special wishes from the Zeiss folks caused some disruption to your daily routines. Again, thanks for having us. Thanks to all the attendees being with us today. Thanks for all the questions. I see that there's a lot of questions that we couldn't answer for time reasons, except one actually that I even can answer. And one of the attendees asked whether this webinar would be ready for on-demand streaming. Yes, it will. And this makes me point you at our website. So the landing page that should be up as we speak, Elyra 7 with Sim². On this website, you'll find a lot of resources. So if you want to learn more about details of the technology and applications, you'll find those there. You'll especially find a white paper there that might help you to review many of the things that René tried to explain to you. And yeah, with this, I'd like to thank you again for joining in. Thanks for being with us today. Goodbye and stay safe.
Goodbye, everybody.
Bye.
Thank you.
Hi, everyone.